It's my pleasure to introduce the next speaker, Adrian Hayday from the Crick Institute in London, bringing cancer immunotherapy to more cancers and to many more people. Just a brief introduction. Adrian started his training at Queen's College in Cambridge and went on to Imperial College London. He then crossed the pond to do postdoctoral work at MIT in the Tonegawa lab, where he worked on Burkitt's lymphoma. He's responsible for describing and characterizing and defining the genes that regulate gamma delta T cells. These are T cells that are involved in sensing tissue integrity as opposed to infection. I think we're going to have a very interesting talk related to cancer immunotherapy. So Adrian, if you could take it from here, that would be great.
Thank you, Eileen. Okay, so first of all, I'd like to thank the organizers for the invitation to talk here today. And what I've heard so far, I've enjoyed a great deal. The work that I'm going to describe today has been done either here in the Francis Crick Institute or here at the Guy's Hospital campus of King's College, London. And I'm going to try and pull several threads of thought together. So, of course, the theme that I was invited to talk to was cancer as a systemic disease. And that's very intriguing to me because the immune system is an established mode for bringing local information to the systemic circulation and taking that information, processing it, and taking it back to the tissue. And this assuredly will be the case in cancer as well. So let's just think a little bit about what we've been experiencing with cancer immunotherapy in the context that I've just described. So I think the last 25 years of cancer immunology and immunotherapy have, in all fairness, utterly revised our view of how the host responds to cancer. Immunotherapy has been a game changer in cancer treatment and management, but it's based on a very important immunological premise, which is that cancers are specifically visible to an immune system that is otherwise tolerized to healthy self. And in that framework, immunotherapy has, to this point in time, revolved almost exclusively around alpha beta T cells. So realizing that there may not be so very many immunologists on the line, I wanted to just communicate a few basic issues. The first is what it is that an alpha beta T cell sees. So what happens, of course, is when a cell gets infected, the viral antigens can be processed to peptides, which are then recognized by highly clonotypic alpha beta T cell receptors. These T cell receptors are immensely diverse because they're assembled by processes of VDJ somatic gene rearrangement, which encode what are known as CDR3s, which are different on every receptor and allow enormous numbers of cognate interactions. And the big realization in cancer immunotherapy and cancer immunology was that likewise carcinogens can generate mutations in genomes that get translated into new proteins from which neoantigenic peptides can be processed. And those too will be recognized by alpha beta T cells. And these clonotypic responses lead to clonal expansions and appropriate effector functions. So these T cells will make strong responses to virus, and they'll make strong responses to mutated self, but they won't make strong responses to ordinary unmutated self because they will have been tolerized both in the thymus and through the action of regulatory T cells. And these expanded cells can assume effector functions that include efficacious cytolytic functions. So this all sounds very good. Now, when T cells are chronically stimulated by antigens like this, they upregulate checkpoint receptors such as PD-1, here's another one, LAG3. And of course, the application of immunotherapy has been transformed through the realization that we could block those interactions or checkpoint blockade. Here's just examples of data. This is now showing that you can combine antibodies against PD-1 and against LAG3 and really get some impressive responses in advanced melanomas, with these curves flattening out here, which suggests that these individuals have generated robust adaptive responses that could be long-term and might actually portend a cure. So what have we learned? We've learned that cancer has an enemy. It's a T cell. It sees it, it kills it, and it may remember it, as I've just said, essentially portending cure. So then the question is, why are we not further along than we are? Melanoma, some non-small cell lung carcinomas, Hodgkin's lymphoma, CLL, but not too many others. And most of what we've seen have been benefits in the developed world. So if we go back to this, we begin to see what some of the problems are. So the generation of these neoantigenic peptides usually requires a high mutational burden on the cancer cell. And there are very many cancers in which that really isn't a feature. In fact, there are some carcinogens that are not even mutagens. So is there a different immune recognition system that might overcome this? The second point is that tolerance issue I told you about. Checkpoint blockade commonly induces severe and systemic adverse events. And those are often auto-inflammatory, even autoimmune responses are driven by reactivity to self, because that single mutation in that peptide can lead through something known as epitope spreading in an inflammatory context to drive the violation of immunological tolerance. So tolerance is fragile. And is there another recognition mechanism that would overcome this? And then the third point is that these peptides, as I mentioned, are presented by MHC. And MHC is often lost from cancer cells, pretty much telling you that the cells have been under immune selection, at least at some point. So it would be nice if we had a non-MHC restricted antigen recognition mechanism. So as Eileen in the chair's role has prefaced, we are interested in gamma delta T cells. Gamma delta T cells are evolutionarily conserved, and their counterparts are even evolutionarily conserved in jawless vertebrates. And we believe that the cells' conserved properties can fill these gaps in cancer immunotherapy, thereby taking cancer treatment to more tumors and to very many more people. And it's because they have a different recognition system. They are not MHC restricted. They have a different mechanism of tolerance. And for good benefit as well, they spend most of the time sitting in the tissues, like the intestine here, human colon here, or human skin, which is where these solid tumors develop. So the way that gamma delta T cells work is completely different to the way alpha beta T cells work. What they do is they use their T cell receptors to recognize surface moieties, proteins, but can also be translational modifications that are overexpressed or misexpressed or misconformed on the surface of infected or cancer cells. The gamma delta T cell receptors can be extremely diverse, and they make very rapid cytolytic responses. So all of this sounds pretty good. And in fact, there is a whole series of ligands that have been identified for human gamma delta T cells, some of which are described here. They will see MHC under certain circumstances or MHC-like molecules. But one particular example of this I'd like to give now is this molecule here, the endothelial protein C receptor, which is recognized by a human gamma delta T cell receptor clone, which was massively expanded in a patient, telling you that although this is a self-antigen, like all of these molecules here, recognition of it immunologically by gamma delta T cells appears to be safe. And the reason we think that is because these basically are not high affinity interactions. For the gamma delta T cell response to occur, it needs to see a lot of something. So even though there will be healthy cells which will express these molecules at a certain level, those for the most part will be beneath the radar. So this is a very interesting recognition. This is EPCR here. Looks a lot like a molecule called CD1. It's actually a lipid-binding molecule, but the recognition of it by this gamma delta T cell receptor does not involve specific recognition of the ligand. It's a highly specific CDR3-driven reactivity. So why is EPCR interesting to the cancer research community? And it's because it's very, very often overexpressed because it sits on chromosome 20 in arm Q, 20Q, and it's part of an amplicon that is frequently amplified because it seems to be sitting in a neighborhood of about 7 putative oncogenes. And it's very, very rarely deleted, which means for whatever reason that it will sit there as a target, presumably because it's just got carried along. So that means this could be a really good target because 20Q amplification is very common. But how can it be a safe target? Is it really just focusing, hinging on this avidity issue that I just described? So I want you to think about these two targets here for the moment when I tell you that the revelation that we were able to make, or experience is probably a better word, in about 5 or 6 years ago, was that the gamma delta T cell receptor, which uses its CDR3 region to bind EPCR, this could equally well be CD1, actually has another ligand. This ligand is known as butyrophilin-like 3, which occurs as a heterodimer with a molecule called butyrophilin-like 8. So where on earth did this story come from? What is it, these T cell receptors having 2 ligands? So it all starts back here with some pictures I showed you earlier, which is that gamma delta T cells have a very interesting association of their V gamma chain with where it is that they sit in the body. So here are mouse epidermal V gamma 5 V delta 1 chains. And we and our collaborators at Yale discovered many years ago now, well, as you see, almost 20 years ago, that a mutation had adventitiously occurred in the FVB strain bred by Taconic Farms that essentially eradicated or ablated this population of cells that can normally be picked up on flow cytometry that they're missing. So together with Rick Lifton, who was at Yale at the time, now at the Rockefeller, we undertook a genetic search, basically, in the old days of classical genetics, to try to find out what the mutation might be. And it turns out it was a mutation in this gene, which we called SKIN1. It was an interesting gene because it wasn't actually annotated at the time, hard to believe. But it's a member of the immunoglobulin superfamily. This is where the premature termination codon occurs in the Taconic strain. But although this was novel, it's a member of a very familiar family because here we've got its NMR structure in orange overlaid over PDL1, the ligand for PD1. And as you can see, they're almost identical. So skin is indeed part of the PDL1 B7 superfamily, but it's actually part of a small family within that superfamily of the butyrophilins or butyrophilin-like molecules. In the mouse, there's a couple of molecules here, BTNL1 and BTNL6, that are counterparts for the human BTNL3 and 8. When we knocked down BTNL1 and 6 in mice, we generated strains here, 3 different strains, that are almost completely lacking these characteristic intraepithelial T cells here that express a V-gamma 7 chain. Of course, we couldn't, by engineering, knock down BTNL3 and 8, but it turns out that the human genome carries an allelic variant that does that for us. And when you look in individuals through colonoscopy, perfectly healthy individuals, you find that the characteristic colonic gamma delta subset that expresses V gamma 4 is almost completely depleted. I won't have time to go into this now, but the significance of this is suggested by a strong association of this genotype with severe Crohn's disease. So let's get back to this. Here we have the capacity to bind a stress ligand like CD1 or EPCR with the very same receptor binding to BTNL3. But it doesn't bind in the CDR3 region. It binds here on a region of V gamma, which is germline encoded. So essentially every cell that expresses this particular V gamma region will be bound by BTNL3 and BTNL8. A non-clonal response is by definition innate immunity. So the T cell receptor, which had hitherto been a paradigm of adaptive immunity, actually has an innate immunity hardwired within it. So we actually, for fun, call these adaptate receptors. But what's also interesting is that these BTNL molecules are expressed on healthy tissue. But if, for example, you have a developing polyp here in a mouse model of APC MIM mouse model, the expression is extinguished. That tells you right away that this gamma delta cell knows the difference between a healthy cell and a transformed cell because its healthy ligand is disappearing, possibly to be replaced by an adaptive ligand. So the cell can pivot in its discrimination of healthy from cancerous tissue, which to our knowledge is fundamentally a completely novel way of tolerance, and thereby gives you a therapeutic window which could significantly reduce the potential for adverse events. So if we go back to that mouse epidermis very briefly, the T cells here are very elegant. They're highly dendritic, and they have the T cell receptor clustered at their surfaces. And if I now remove the mask and show you where the epithelial cells are, here the T cell receptors are binding to these epithelial cells at steady state. And in fact, we can image the V gamma 5 receptor with the SCINT1 gene I mentioned to you in super high resolution microscopy. So what Duncan McKenzie did was simply take a perfectly healthy mouse and introduce into the skin an anti-SCINT1 antibody to block that interaction. It had a profound impact. The skin began to swell. The animals are unable to maintain their water balance. Transepithelial water loss increases. And that's because the keratinocytes aren't working anymore. They're losing their expression of molecules like transglutaminase, loroquine, and IL-13. So this says that these steady state interactions are occurring in healthy tissue all the time, and it has a profound contribution to physiologic maintenance. So this pivot says under normal circumstances, you have this really key physiologic interaction. The cell is engaged in an active way. It's tolerant to self because it's contributing to self. When everything changes, the cell now can adopt its clonotypic response to ligands such as EPCR it can do it safely without having to worry about it targeting on a normal cell. So let me just finish by saying, okay, sounds all right, but is there any actual evidence in support of this? This is just a list. It's not wholly comprehensive. It was a list that we put together when we published this review last autumn. And it just lists an extraordinarily wide range of ways that people are using gamma delta T cells in the clinic. And they're all showing very strong safety profiles. So that sounds good. Secondly, there's very many natural assumptions of human gamma delta T cell populations with possible positive outcomes in cancer, consistent with our original publication in 2001 that gamma delta T cell deficient mice are susceptible to cutaneous cancer. And finally, any application to solid tumors will almost certainly need to be in combination with PD-1 blockade as standard of care. And what we were able to show, and published about a year ago now, is that actually when you look at those associations of gamma delta T cells in the skin with much better outcome in individuals being treated with anti-PD-1, so these are melanoma patients under checkpoint blockade, the strongest association is with tumors of low neoantigenic load, which would essentially be very poorly visible to alpha beta T cells, which really need that high neoantigenic display. So this is my final slide. Alpha beta T cells have been doing a great job. MHC loss turns off the light. They can't see them anymore. They end up synonymous with low mutagenic burden tumors like pancreatic. But thankfully, gamma delta T cells are there to see them. And some of those targets can be neutral targets amplified in very many cancers that at this point in time we're having trouble treating. So my hope is to be optimistic and take these sort of ideas forward in order that molecules like this can indeed be safely targeted by gamma delta T cells. So I'd like to thank you for your attention. This is the group. I haven't really mentioned anything that's unpublished, so you can find your names on all the papers. What was unpublished was trying to synthesize these ideas together in the context of this very nice meeting that you've put together. So thank you very much.
Well, thank you, Adrian, for that marvelous talk. Maybe I could start off the questions by, is anyone deploying this knowledge, like the presence of gamma or absence of gamma delta T cells or the butyrophilins for, diagnosing cancers?
Yeah, so it's a very good question, Eileen. It's early days, particularly for the butyrophilin-like molecules, because serological reagents have, they've taken a while to develop. But there's some beautiful work being done, not in the systems that I described today, but actually in relation to human peripheral blood gamma delta T cells and hematological cancer targeting by Daniel Olive's group in France. And they certainly have very nice reports appearing now looking at associations of disease with different status of butyrophilins, for example, because there's not only changes in levels of expression that you can monitor, but there are actually allelic variants, just as I described today. So, there's plenty of things to look at.
Great. We have a question from an attendee.
Yeah.
Can tumors be hot/cold in regard to gamma delta T cell infiltration?
Yeah, very much so. And there's at least 3 situations in which the presence of hot gamma delta T cells has correlated very, very well with patient responses. One is in triple negative breast cancer that was published in Science Translational Medicine in 2019. In 2022, in Nature Cancer, the association with non-small cell lung carcinoma. And that was backed up by some very good TGCA data and ovarian cancer paper out of the Karolinska Institute. I'm tempted to think that was Science Translational Medicine as well, somewhere around that time. So those papers exist if you're interested in following it up, but they're certainly very very indicative clues that this is at least going to be a helpful way to go forward.
Well, great. I think we need to move on because of the time.
Sure.
That was a wonderful, informative talk.
Thank you.
And